So our lab is mining cancer genomes to try to find interesting new ways of treating cancer by understanding how these mutations in DNA are affecting cells. And so the process that we primarily look at is through alterations in RNA. So RNA is sort of the output of the genome. Broadly, our lab is looking for these ways that cancer genomes get mutated that then disrupt the way RNA is produced. For our Giving Day project, we have 4 talented graduate students in the lab.
So I'm Cameron. I work in Angela Brooks' lab. What we generally look at in cancer research is how genes are regulated and how genes are expressed. How DNA is converted into this RNA, and these RNAs are kind of like messages that code for very specific and important functions in the cell, like growth and proliferation. What we do in our lab is we sequence out these messages using this new technology called nanopore sequencing. We can compare that cancer-specific message to a normal cell message and see how it's changed in these 2 different environments. So with the Giving Day money, I'll be using it mostly to purchase flow cells for my experiments, plus the library preparations that are associated with those flow cells, and all other necessary reagents that I'll need for the RNA extraction and cell culturing.
We know that cancer arises because of mutations that are occurring in our genome, but one of the things that we don't really know about is how mutations in these sort of non-coding regions are affecting translation. So I'm trying to basically develop a new computational method to investigate these kind of unknown mutations that are occurring in the 5' UTR, because I'm trying to develop a variant caller pipeline that's massively parallelized. I'd like to raise money to basically fund some compute time. Right now I'm kind of estimating it would probably take about $25 an hour to run my pipeline on an 8-core machine. So it'd be great if I could get a ton of hours to run multiple cores simultaneously.
Hi, my name is Brandon, and I'm in the Brooks lab. My research focuses on developing a new method to map nucleosome positions. Nucleosomes are what cells use to package DNA, but where their position is also important for gene regulation. I'm developing a new method to modify DNA and use nanopore sequencing to determine chromatin structure. Your money would go towards paying for reagents that cost $50 to $200 per sequencing run, and flow cells that cost $500 each.
Mutations in RNA-modifying enzymes have been implicated in cancer. My research aims to investigate the connection between RNA and RNA modifications in the context of cancer. And so the best way for us to look at this would be using direct RNA sequencing. Direct RNA sequencing allows us to detect both RNA modifications as well as the whole transcript. This project can really push forward our understanding of RNA modifications in cancer in a way that no one has really looked at before. However, in order to complete this project, I would need help funding the cost of flow cells and other reagents. Every contribution would be immensely helpful, so please consider donating. Thank you.